Hi everyone, very glad to join you here from my maternity leave and to tell you more about how we use our neighbor labeling system PUFFFIN to illuminate cellular neighborhoods in model systems of development. So I work in cellulose lab at the University of Edinburgh, and we've been very interested for quite some time now in how changes in cells influence their neighbors during development. So if cells, for example, decide to undergo a certain differentiation, how will they communicate that to their neighborhood? And for example, would they tell their neighbors to do the same as them, resulting in kind of local synchronization? Or maybe they do exactly the opposite, telling their neighbors to not undergo the same differentiation, resulting more in a salt and pepper kind of pattern. Or it could, of course, be that there's just no influence on neighbors when changes in cells happen. And which of these mechanisms you have is probably highly dependent on what event of development you're looking at, which mechanisms are at play there. So in recent years, a part of Sally's lab has focused more on developing new tools to look at these potential neighborhood effects and to find ways of identifying the local neighborhood of cells of interest. And one of these systems I was working on as my PhD project, and the idea was that the cells of interest send out a fluorescent protein to the neighbors, allowing us to isolate the cellular neighborhood. And how we go about this is we're using a highly positively charged green fluorescent protein. And if that gets out of the cells, it can then interact with the negatively cell membranes of cells nearby, and through that charge interaction, that then triggers uptake into the cells. And because all cell membranes are negatively charged, we have a really universal process here. So the supercharged GFP also has a secretion signal, so that it gets out of the cells that are expressing it. And to distinguish between the labeling cells and the label cells, we always need a second fluorescent protein in a different color. And in this case, for example, we can use a red fluorescent protein. One more thing that I have to mention here is that the supercharged GFP on its own is actually not that bright. At least for our purposes, it wasn't bright enough. So we always fuse it to an amplifier, and that could either be a very bright green fluorescent protein like M neon green, or it could be the halo tag that itself does not have any fluorescence, but you can add fluorescent ligands in any color you like to your culture, making the label detectable when they bind specifically to the halo tag. So that's kind of how the system works. And if we have these two fluorescent proteins, we're able to tell apart three different populations. So we'll have our double positive secretors, that will be your cells of interest. We then have their neighbors that become labeled, so single positive just for the label color. And then we have cells that were too far away to receive the label, and they just remain double negative. And if we can identify these three populations, for example in flow cytometry, then we can also sort and isolate them, allowing us to do, for example, gene expression analysis or even functional assays with the live cells that we sorted. And another great feature of this system is that all components required to set it up are actually contained in a single plasmid. And so you can just take this plasmid and introduce it to your cells of interest, and there are no requirements for the receiving cells, so the potential neighbors. And this is because we're using this modular EMMA cloning strategy, and I'll briefly talk about that or come back to that a little later. But it's kind of like Lego bricks, so we can assemble lots of different versions of this plasmid. And we call the system positive ultra-bright fluorescent fusion for identifying neighbors, because that's what it is and that's what it does. And that stands short for PUFFFIN. So does it work? So here we have, as I mentioned just now, we have unmodified cells that are double negative for the label and the nuclear marker, because we've not transfected them with anything. Then we have our secretors that are expressing both these proteins, and we can see they're very bright double positive for these two. If we mix them just before analysis, we can see that they're very well separated in flow cytometry, but if we leave them in a core culture, in this case for 48 hours, we see that all of these unlabeled cells become positive for the label here, but not for the nuclear marker. So this is really great. It shows us that it works. But can we call this a neighbor labeling system? No, we have to show that we're really just labeling the cellular neighborhood. And we've done a lot of extensive and careful characterization of this. So please have a look at our paper if you want to know more. But I just want to show you my favorite picture from the paper, and that's this one here. And here we're using the halotag version of the system together with the far red ligand, allowing us to do really nice live imaging. So these are live cells, and you can see here in the middle, in pink, are our secretors with their nuclear marker, and then around them is this really nice cloud of label here in amber, but not further away. And especially looking at the side views here, we can see that the label actually does go into the neighboring cells, and it's not just stuck to the outside. So we're really happy with this, and quite convinced that it works the way we want it to work. And then a great or we've also done a lot of characterization on timing, so we know that it's really fast. So depending on the context, you can get really nice labeling within the first hour. So we get really fast and really bright neighbor labeling here. And another great feature of the system is this modular cloning strategy that we're using, because that allowed us to also very easily construct minimal PUFFFIN cassettes, and they're small enough that we can deliver them in vivo. So this experiment was done by Julia, our collaborator at the CRIC, and she used one of the minimal PUFFFIN constructs and electroporated it into chick embryos, and you can see this looks very similar to the cell culture picture I just showed you, where you have a secretor in the middle and then this cloud of label around it. And then another collaborator who's like us from the University of Edinburgh, Lida, she used the lentivirus that we made with the minimal PUFFFIN construct and injected that into mouse cortical brain slice cultures, and you see that looks very similar to the two pictures that we just looked at. So we're quite happy that it can work in vivo, but I'll just really quickly want to tell you about an example of how we can use it, and that's what I've been working with for the past year, and that is looking at the exit from naive pluripotency, and we want to know if cells synchronize the exit from naive pluripotency with their neighbors. So we're using mouse embryonic stem cells that are expressing the PUFFFIN construct, and we're giving them a head start to differentiation by kind of pre-differentiating them for 24 or 48 hours, and then putting them in a core culture with naive unmodified cells, and then we ask in a colony formation assay where only pluripotent cells can form colonies, is there a difference between the neighbors and the non-neighbors from the same dish? So we can just count colonies and see how many pluripotent cells we had there, and we see that in a control condition there's not really much difference between neighbors and non-neighbors, so they form the same amount of colony, but when we look at neighbors of pre-differentiated cells, we see that actually they form fewer colonies, so we have less pluripotent cells compared to non-neighbors from the same dish. So we're really excited about this. This kind of suggests this local synchronization in this case, and we're doing follow-up experiments on that now. So I hope I could just show you that PUFFFIN is a really great system, and we're so excited about seeing what other people might do with it. But I just want to really say thank you to all the people who supported me on making this possible. So thank you for listening, and I'm happy to take questions.
Thank you so much, Tamina. Does anybody have any questions that they want to type into the chat? Maybe while you're thinking, I'll kick off, Tamina. So what sorts of time frames do you see this operating in? I'm particularly interested in how you could use this in vivo, I guess. But of course, in vivo cells are dividing, they're proliferating, they're moving around relative to one another. So I was interested to know about really the timing of how quickly you saw the response, I suppose.
Yeah, that's a great question. I actually have a backup slide. So this is live imaging from an in vitro culture, and we're taking a picture every 15 minutes. And I'll just go back and show this. Sorry, again. So within the first 15 minutes, we already saw labeling appear around
Wow.
them. It really depends on what you're looking at, and it also depends on how many secreting cells you have as your neighbors. Obviously, if you have more neighbors, then we can detect the label much quicker. But so, for example, when we use this 4 secretors to every 1 unmodified cells, we saw like really clear detectable label within 15 minutes as well. If you're doing live imaging, that's of course possible as well. And the longer you wait, the more labeling you get. But we kind of hope that in vivo you can maybe see the trace of cells where
Yeah,
when they walked by and labeled the cells that they came
fantastic.
across, and we definitely think that's possible.
Excellent. Thank you so much. I don't see any more questions in the chat, but if you do have further questions for Tamina, oh no, Stan does have one. So we'll just take this one last question. Have you been able to add single puffin cells and see how many cell bodies it travels? Yes.
Yeah, so I think this is more what we've done here. I mean, it's not single cells, but we kind of created this boundary between unmodified cells and secretors. And on this experiment, we could then do a bit of quantification on how far the label travels, if this is kind of your question. And what we saw there is that obviously the M cherry falls really quickly, our nuclear marker, so this is our last secretor cell, and then we see this kind of gradient of the GFP label, and at about 3 cells away for these cells we had the half maximum, where we see you can get still nice detectable label. So this is kind of the closest we got to answering that question. It's really hard to do, so I hope this is what you were asking for.
Thank you so much, Tamina.